All right, guys, if you were absent on Friday, you've got a couple of notes to make up. I'll try to make this video as fast as I can, but I want to say please bear with me. I'm trying to use two different tools. I'm trying to use a screen recording tool and the Google Slides tools at the same time, and it's going to get a little bit confusing for me, so just bear with me. All right, so anyways, Punnett squares. Make a new section in your or a new table of contents entry and a new blank page for this. They relate to the notes that we took yesterday on Thursday about Gregor Mendel and his peas, but they are a little bit different. It's a slightly different topic, so I want a new section in your notes for them. All right. So you don't have to take any notes on this slide. I just want to quickly talk through it. So yesterday we talked about how Gregor Mendel did his studies in the mid 1800s, and we also mentioned how his studies weren't really appreciated in his time. It wasn't until later on that the data he collected was really seen as important. Well, this is sort of the time period that we're jumping to. At this point, Gregor Mendel is dead, and this dude, Reginald Punnett, who was actually a statistician, started to find his data and do some more things with it. Now, real quick, Reginald, I'm going to skip way backwards in the slideshow here, so bear with me. Reginald Punnett was a statistician, okay, and he was asked to explain why is it that this white flower pops back up after 2 generations. It kind of makes sense that if you have parents of 2 different colors, maybe all of your children would only be one of those 2 colors. Like, okay, that's okay. That's maybe understandable. But what's not understandable is how 2 purple parents can all of a sudden start producing 25% white children or white flower children, you know? Reginald Punnett was asked to help explain that. And he wanted to make sort of a visual representation of what was going on. And so he came up with this thing that we now call a Punnett square, which is this. Again, please don't try to copy this down into your notes. It's just so that you can start getting an understanding of what a Punnett square is and how it works. So for one, the parents' alleles go on the outside of the square. That means that these are parent alleles and these are parent alleles. Remember, we all have 2 alleles for each of our genes. We all have 1 chromosome that we got from mom and 1 chromosome that we got from dad, and they all contain, each chromosome contains alleles for the same gene. Okay, so you may have gotten a brown eye allele from dad and a green eye allele from mom, and the trait that you express is some mixture between those 2, right? So anyways, mom or dad has 2 alleles to contribute. Mom or dad has 2 alleles to contribute. It doesn't matter if mom or dad goes in whichever spot. So parents go on the outside, and therefore the possible combinations of alleles that you can have in the offspring go on the inside. Now think of this kind of like a multiplication table. You guys have probably all worked with multiplication tables. Say you want to know what 2 times 3 is on a multiplication table. You're going to find 2 on one side and 3 on the other, and you're going to see where they meet in the table, and you're going to see a 6 there because 2 times 3 is 6, right? So a Punnett square works similarly. If this parent contributes this gene, follow it to there. If this parent contributes this gene, follow it to there, and then you would see the combination trait or the combination of genetics that the baby would have. So anyways, super quick, brief introduction to what a Punnett square is and how it works. If you're confused, it's okay. We're going to continue working with these, and they'll make more and more sense over time. All right. So let's keep moving on. I do want you to take some notes on probabilities. So for one, the probability of something is the likelihood of a particular event occurring. So this should be going in your notebook because this will be on a vocab card. Probability is the likelihood of a particular event occurring. Now, it can be expressed as a fraction, percent, or ratio. Mr. Lord has told me that you guys focus mostly on fractions, but I also need you to know how to represent certain things in percents and in ratios, and we're going to get some practice doing that in a second. Pause if you need to, but I'm going to keep going. So fractions shouldn't be too difficult, and really we're only working with a 4-piece square here. There are Punnett squares that are bigger, but we are probably, for the most part, going to stick with this. It's only going to be like extra credit or something if you have a big, giant square. So we want to talk about ways to represent how many offspring are purple. Let's represent how many offspring are purple as a fraction. Okay, so a fraction would say how many offspring are purple compared to how many offspring are there total. So we have 3 purple offspring and 4 offspring total, right? So that means that our fraction is 3 out of 4. Okay, I could ask you the same thing about white flowers. How many flowers are white out of how many flowers total? Hopefully your answer would be 1 out of 4. Then you have 1 white out of 4 total. Okay, for percentages, it's basically the same thing. Out of your 100% of all offsprings, what percentage of them are purple? Now, you may already just kind of know that 3 out of 4 is 75%. If you are shaky on that or you want to double check yourself, type this into a calculator. If you do 3 divided by 4, you'll get 0.75. So that'll tell you that your percent is 75%. Similarly, if I were to ask you the opposite, what is the percentage of white offspring here? You would tell me 25%. And if you weren't sure how to get that, but you are comfortable with fractions, you can say, okay, well, 1 out of 4, and you can divide 1, you can do 1 divided by 4, and you will get 0.25, which is 25%, right? So you'll get used to it. And there's really only a handful of percents you got to know. 0%, which should be pretty easy. That means none. 25, 50, 75, and then 100, which is all. So really, there's only those 5 that I need you to know. Finally, we come to ratios, which students are going to get mixed up sometimes with fractions, so be careful. Now, a ratio says how many of this thing to how many of that thing. And the 2, meaning TO, not the number 2, but a ratio of this to that 2 is represented by a colon, so 2 dots on top of each other. So in this case, we want to know how many purple to how many white, all right? So we have 3 purple to 1 white, which means our ratio is going to look like this: 3 purple to 1 white, all right? So if I were to ask you the same thing, but inverse, how many white to how many purple, your answer would hopefully be 1 to 3. 1 white to 3 purple, all right? Hopefully you can handle this. I'm sure that with a little practice, you'll get it without too much trouble. You just got to keep these straight in your head. And it might be helpful to write this little table down and at least have one example of each thing so you know, oh, shoot, she's asking for a ratio, but I forget what a ratio looks like. Oh, okay, it's the thing with the colon in the middle. Now I remember. Now I know how to do it. So this table might be a helpful thing for you to copy down, even if it's without this part up here. Whatever you feel you need to do, you decide for yourself, all right? Moving forward, we're going to quickly talk about how to use a Punnett square. I'm going to see if I can do this on here while I'm talking. This is where you need to bear with me because I'm trying to use multiple tools at once. So step number 1 is to draw a box with 4 boxes inside like a window. What I mean by that is just what these Punnett squares have been looking like so far. Okay, so there's your box with 4 boxes inside like a window. When I say like a window, I mean like that's how you would draw a window on a little cartoon house if you were drawing a house for a stick figure. All right, hopefully we can make this work. Go away. I need you to go away. I need you. Well, there we go. Okay. So then your next step is to place parent alleles above and to the left of the square. Now we already kind of saw this earlier. Also, your parent alleles are going to go up here and over here. So above and to the left. I'm sorry, I should have said this already. I don't think you need to write this down in your notebook. If you're already not writing this down, great, that's fine. You will get used to this very quickly, and it's not going to be a problem for you. All right, let's move on. Okay, all right. So then distribute the alleles down and to the right in the boxes. So what I mean is you'll have 2 alleles up here for one parent, and you'll have 2 alleles over here for the other parent. And all you need to do is take the allele here, distribute it down, take the allele here, distribute it down, take the allele here, distribute it to the right, and here, distribute to the right, okay? Once we see that in action, it'll be pretty simple. And we already said that the 4 inner boxes represent the possible offspring allele combinations, right? So let's see if I can delete that. That didn't work. All right. Don't mind Miss Richard. She doesn't know what she's doing. Boop, 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 boop. All right. Before we go any further, you got a little bit of vocab that I want you to write down. Remember, you really need to keep up with vocab in this unit. If you don't, you're going to get super lost. New word, genotype. A genotype is the genetic makeup of an organism represented with big and little letters. If that's confusing to you right now, that's all right. We're going to talk about it in a little bit. The other thing we're going to talk about is phenotype. I'm sorry, I'm trying to play with these tools and talk at the same time. Phenotype and genotype are words that generally go together. If a genotype is your genetic makeup, a phenotype is your physical description. So think genotype, genes, phenotype, physical. All right. Let's look at a couple of examples here. So first off, we've got this one. I'll have 3 pictures of flowers in the end. They all have a couple letters by them. Those little letters there are their genotypes. They're represented by big and little letters. Now, this one's a little bit weird. It's a W, so it's hard to tell if it's a big or little W without reference to anything. So I'm just going to tell you that it is a big W. So if we're talking about what is the genotype and phenotype of this flower, the genotype is going to be big W, big W. Now, we could say this capital W, or, uppercase W, but that's so many more syllables that you have to say. So just say big and little, and you'll be fine. Okay, so big W is the genotype. The phenotype, the physical description, we're focusing on color here. So the phenotype for this flower is just purple, right? Looking at the next one, here we can tell the difference between big and little w's. Now this one's genotype is going to be big W, little w. I want to take a quick pause here for a second. Now, if you are, I know we do a lot of typing in my class, but if we are ever doing anything where you have to write a genotype like big W, little w, I want you to make sure that your letters are clearly bigger or littler. So, for instance, if you were trying to write big W, little w, and you wrote this, well, I can kind of tell that second one is a little bit smaller, but it's not smaller by very much. So what instead I would want you to do is write your big W pretty big and your little w so small that it could not possibly be mistaken for a big W. So make a very clear distinction between your big and little letters when you are doing these Punnett squares. All right. Continuing to move on, if I can get to there. All right, the phenotype for this one then is just going to be, again, its color, which is purple. For this last one, the genotype, the genetic makeup, we would say this one, little w, and its phenotype this time is going to be white. All right. Oh, darn it. I was hoping that would go away. There we go. All right. So we're going to be doing some drawing here. What I want you to do in your notes is draw yourself a square like this. So we're going to start doing your first Punnett square. Now, we want to do a cross between big P and little p is what it says up here. And the question is, what percent of the offspring would you expect to be purple? Well, in order to find that out, we need to actually do the square first. So we're going to start out by drawing our parent alleles. My first parent is big P. I'm just going to draw that one on the top. Doesn't matter where it goes. Okay. Big P on the top there. And then I have little p that needs to go on the side. It's going to be a little bit difficult to draw little on the screen, but that'll do. Okay. So now I need to fill out my Punnett square. I need to take the letters on the top and drop them down into the box, both of them. And I need to take the letters on the side and pull them over. And I'm going to do one box at a time. So I'm going to start with this upper left box here. So if I start there and I drop down my big P, and then I pull over my little p from the side. There we go. Again, multiplication table, combining a big and a little in that box. Okay, upper right-hand corner, we've got big P coming from the top and a little p coming from the side. Coming from the top in the lower left, we've got a big P, and coming from the side, we have a little p. And what do you know? Big P and little p in our last box as well. So this is what your Punnett square should look like in your notes right now. I do want this to be in your notes, so you have some examples there. Now the question is, what percent of the offspring would you expect to be purple? Well, we have to remind ourselves then what the I don't know how I feel about that. Oh. What? Sorry, I'm trying to understand what's happening with my mouse right now. I don't like it. Go away. Oh. There it is. Okay. So, sorry. Told you to bear with me. So we have to remember what these different letters mean and what they mean for what the flower looks like. So this big P is our dominant allele. If you remember from yesterday, we said if you have the dominant allele, if you have a big letter, that trait is going to show up. So in any of these where we have a big P, it's going to be purple. If there are any that have no big P's, oops, just accidentally went to example 2. If you have no big P's, if it's just little p's, that's when you show the white color. Okay, so in all of these examples though, all of them have at least one big P, which means that all of them will be purple. So our answer then to this, and I want you to write this down also sort of off to the side, is because we were asked what percent of the offspring would be purple, we want to say 100% will be purple. Or 100% purple. All right? So let's clear that off, and we're going to go to the next slide. Hopefully, Miss Richard can figure out how to do that. Boom, boom, boom. All right, here we are at example number 2. Okay, we're going to do the same thing again. But now we have different parents. We have a cross between big P little p and big P little p. So again, we want to draw our letters over here. By the way, let me make a quick note here. See this big P little p? See how the big P comes before the little p? That is always how it's written. Some of you may know that my first name is Cassidy. Now, what would you think of me if I wrote my name like that? That looks kind of dumb, right? That's not how you capitalize names. That's also not how you capitalize genotypes. When you write a genotype, the capital letter always goes first. Do not put the capital letter second. You're not going to be entirely wrong, but that's just not the way that it's ever written. Make sure the big letter comes first. All right, so anyways, we're going to do this cross. Big P, little p is one of the parents. So I'm going to write big P, little p up here. All right. And big P, little p will go on the side as well. Big P, little p. All right. So let's go ahead and fill in this square. If we look at the top left square first, we pull down a big P and we pull over a big P. In the top right square, we're going to pull down a little p and we're going to pull over a big P. Notice how I wrote my little p off to the side because I knew that I was going to be drawing a big P next to it. Okay, in the lower left, we've got a big P and a little p. And finally, we've got a little p and a little p. All right, so now we got to come back up to the question. There's our completed Punnett square, and the question asks, what percent of the offspring would you expect to be white? Again, remember that anytime this big P shows up, it's going to be purple. Doesn't matter if it has 2 big P's or only 1 big P. That big P being there means that the flower is going to be purple. So this upper left is going to be purple, upper right is going to be purple, lower left is going to be purple, But this lower right is the only one that doesn't have a big P. It only has little p's, which means it is going to be the only one that is white. So what percent of the offspring would you expect to be white? Only one of these four squares would be white, so that means 25% white. Okay, now, with these last two examples, maybe it reminded you of a certain monk that we studied, right? We started, let's go ahead and erase this. If we go back a slide, hopefully we're done with this for a little while. All right. Yes, if we go back a slide, we'll see we started with big P, which would be purebred dominant, purebred purple, and little p, purebred white. Okay, purebred purple crossed with the purebred white. Hold on a minute, Gregor Mendel did that. How many purple offspring did we get? What percent of the offspring were purple? 100%, just like Gregor Mendel found. Now, we take two that are across. Remember, in example number 1, all of the babies here were all big P, little p, right? In example number 2, we're taking two big P, little p's, basically the children from example number 1, and we're crossing them. What percent did we find to be white in that one? 25%, just like Gregor Mendel found in his studies. So the Punnett squares that we just did were visual representations of Gregor Mendel and the studies that he did. We represented graphically what he just looked at, looking at his flowers, right? So, kind of cool. We're going to do a couple more examples shortly, but I want to get a little bit more vocab first. Sometimes we describe genotypes based on the combination of alleles or letters they have. Homozygous means having two copies of the same allele, and we can have that in two different forms. We can have homozygous dominant, which means two of the same big letters. So it doesn't matter what letters, as long as they're both the same and they're both big. And then homozygous recessive, same letters, but they're both the small ones. Okay, and then heterozygous means having one of each allele. So you'll have one big letter and one little letter. That would be heterozygous. Now, something that I compared this to in class, most of you guys know the term homosexual. Homosexual people are attracted to people of the same sex. Homo means same, having two copies of the same allele. Heterosexual, in other words, straight. Heterosexual people are what we consider straight. The heterosexual people are attracted to people of a different sex. Hetero means different. If hetero means different and homo means same, that might help you remember which one's which here. Now, with that being said, this became an issue in class. These alleles are not gay. These alleles are not straight. Gay and straight pertain to homosexual and heterosexual. All right? These are not homosexual and heterosexual. They are homozygous and heterozygous. I don't want to hear about genes or alleles being gay or straight. That's not what it is. All right? So, just gave you those terms to help you remember maybe what they mean. Homo means same. Hetero means different. Moving on. Let's get a couple more example crosses done here, which Miss Richard loves so much. All right, so this time we've got a cross between big W, little w, and little w, little w. So big W, little w, little w, little w. Pull down a big W and pull over a little w. Pull down a little w and over a little w. Down big, over little. And then here we've got, I almost made that into a V, two littles. All right. So there's our completed Punnett Punnett square. And then the question says, what would you expect to be the ratio of heterozygous to homozygous? So remember, hetero means different, and we're talking ratios here. So if we're talking ratios, we just want to count this to that. All right, heterozygous to homozygous. So let's find our heterozygous first. Hetero means different. These two right here, big W, little w, are the different ones. So that would be 2 hetero to how many homo? Over here we have homozygous. We have 2 of them. So we have a 2 to 2 ratio, which we should see simplified as 1 to 1. Okay? Let's get rid of that. Move on to there. I can move this. Nope. All right. Okay, so now we've got a cross between big S, little s, and then big S, big S. Upper left square, pull down a big and over a big. Upper right, we'll pull down a little and over a big. Lower left, down a big, over a big. And lower right, down a little and over a big. All right. So then we want what fraction of the offspring would have a heterozygous genotype. So we're looking for, if it's a fraction, a number over a number. So total, we have 4 offspring. And we want to know how many would have the heterozygous genotype. So of these 4, how many are heterozygous? Well, these on the left-hand side are both have the same allele. These guys on the right-hand side have the different alleles. So 2 are heterozygous. 2 out of 4 we can simplify to be 1/2, right? So don't want to see 2 out of 4. We just want to see 1/2. And there we go. All right. One last example, and then I'm going to let you go. There we go. All right, now we're going to do a cross between big Y, little y, big Y, little y. Okay. Top left, pull down a big, over a big. Top right, down a little, over a big. Bottom left, down big, over little, and bottom right is 2 littles. All right. So there's our Punnett square. Question is then, what will be the ratio of yellow to green seeded plants? So we're looking for a ratio. So we want to count our yellow to our green. All right. So first off, we got to figure out how many are yellow. Remember, anything with a big Y is dominant. Anything with a big Y is going to be yellow. If I don't have the big Y, that's the only time I'm going to be green. So I have 1, 2, 3 that have at least 1 big Y, and I only have 1 that is all little y's. So 3 to 1 is our ratio, just like we saw earlier in our notes, right? So if you've got this in your notes, if you feel, well, really, if you've got everything in your notes, great, you're done. If you have any questions for me, please let me know. Just make sure this gets in your notebook. And don't be afraid to ask me questions.